course. Hi, everyone. So I'm Elliot Rees. I'm a research fellow at Cardiff University, and it's my pleasure to introduce Sophie Check. So Sophie is a second-year PhD student, supervised by myself and Professor James Walters, and her PhD focuses on using exome sequencing data to identify new genes that have a role in schizophrenia. So with that, I'll hand you over to Sophie. Thank you.
Thanks, Elliot. As Elliot says, I'm a second-year PhD student at the Cardiff University Centre for Neuropsychiatric Genetics and Genomics, working in the psychosis group. My research interest is in the genetic causes of schizophrenia and other psychiatric disorders. And my PhD project focuses specifically on using rare genetic variants to identify schizophrenia risk genes. So schizophrenia liability, which is the likelihood of developing schizophrenia, is influenced by genetic risk factors and environmental risk factors. And of these, it's estimated that genetic risk makes up 60 to 80% of variation in schizophrenia liability across the population, with environmental risk factors like early adverse experiences making up the other 20 to 40%. And these genetic and environmental risk factors increase predisposition for schizophrenia, but they don't guarantee that someone's going to develop it. For example, if an individual has a strong genetic risk factor, it doesn't mean that they'll definitely develop schizophrenia, but it puts them at a higher risk and means that they might be less resilient to environmental risk factors. And the useful thing about genetic risk factors is that identifying them can help us to understand the biology that's disrupted in schizophrenia, and it can also potentially help us to understand how we might go about treating it. So these genetic risk factors can be broken down into common variants, which are found in more than 1% of people, and rare variants found in less than 1% of people. And rare variants generally have much larger effects on schizophrenia risk than common variants, which are common in the population because they don't have strong impacts on schizophrenia risk. And rare variants also impact genes more directly, in contrast to common variants where it's difficult to know which variant is having an impact. And as a result, rare variants can be used to implicate specific genes and biological processes that are involved in the development of schizophrenia. And in order to implicate specific genes, we basically sequence genes in a large group of people with schizophrenia and a large group of people without schizophrenia, also called whole exome sequencing. And this can either be a case control sample where the cases and controls are unrelated, or a trio sample where you're comparing affected children to their unaffected parents. And in case control samples, these gene sequences represented in grey are compared to look for genes where rare variants represented in red are associated with having schizophrenia. So the top set of genes shows all the rare variants that are found among cases, and the bottom set of genes shows all the rare variants that are found among controls. And if there are significantly more variants in cases, then we say that that gene is associated with schizophrenia. In trios, it's slightly different because you're looking for variants that are found only in the affected children and not in the unaffected parents, but otherwise the principle is similar. And one of the consistent findings from these rare variant studies is that rare variants are enriched in genes where damaging variants are more likely to be harmful, called constrained genes. And this is a set of genes that would include genes that increase risk for schizophrenia. So this is what we would expect to see. This is shown in the figure on the right. So a study in 2017 compared 4,000 cases to 9,000 controls and 1,000 affected children to their unaffected parents. And they found that rare variants were enriched among constrained genes, but not among other genes. And that this enrichment was driven by rare variants with damaging effects. So we can implicate this set of constrained genes by comparing to cases and controls on this scale. But what we ideally want to do is to narrow down this association to specific genes that are involved, which requires much larger sample sizes than this. And this was achieved in a landmark study published last year by the Schema Consortium, where they meta-analysed or combined all existing schizophrenia case control and trio samples along with new whole exome sequencing data. And in total, they compared 24,000 cases to 97,000 controls, as well as 3,000 trios. And with this sample size, they managed to significantly implicate 10 genes, which is a huge step forwards. Significance here refers to multiple testing significance. So there are about 21,000 genes in the genome. If you test all of them, you expect, say, 5% of them to be associated just by chance. So to adjust for that, we impose a significance threshold, which is shown as a red dotted line. And there's a less stringent threshold shown in green underneath. And the genes that are associated above this threshold give interesting insights into schizophrenia biology in the study. So for example, GRIN2A and GRIA3 are both glutamate receptor subunits. So this supports the hypothesis that glutamate signaling is disrupted in schizophrenia, and this might be a potential therapeutic target. But there are many more genes involved in schizophrenia than just these 10. We're really just starting to scratch the surface of gene discovery and what it can tell us about the underlying causes of schizophrenia. And to implicate genes in greater numbers will require larger sample sizes still. So to contribute to this gene discovery effort, a new schizophrenia case control sample was exome sequenced at Cardiff. This is made up of 4,661 cases and 5,713 controls after processing and quality control, which was carried out by my supervisor, Elliot. And the objectives for analysing this new sample were firstly to evaluate rare variant enrichment in this sample and whether it's consistent with and supports findings in previous rare variant studies in schizophrenia. And secondly, to meta-analyse this sample by combining it with the schema sample in order to expand the sample size and increase power to detect associations. By combining the sample with schema, we expand on the number of cases in schema by 19% and increase the total number of controls to more than 100,000. So this gives us a substantial increase in power to potentially implicate novel genes. So firstly, looking at rare variant enrichment, we replicated the finding that constrained genes are enriched for rare variants. So this is reassuring because it shows us that this sample has the profile of rare variant enrichment that we would expect to see, and also that it's consistent with other schizophrenia case control samples, and therefore we can be confident that it's appropriate to meta-analyze it with other samples. And we also found that the 10 schema genes and the wider set of 32 schema genes are enriched, which is also really encouraging because it independently supports the involvement of these genes in schizophrenia. And it suggests by combining these samples, we might be able to strengthen these associations. So we then combined this new sample with the schema sample to give a total of 28,909 cases, 103,035 controls, and 3,444 trios. Sample, again, looking at in red, we were pleased to see that we replicated the majority of the schema genes, again, providing independent support. And we also implicated 3 novel genes, which are shown in bold. So these are STAG1, ZNF136, and MAGE-C1. And these novel genes are interesting because they give insight into how the biology of schizophrenia overlaps with other psychiatric disorders. So STAG1 has previously been implicated in intellectual disability, and MAGE-C1 is also an autism risk gene. Which lends support to the theory that the genetic architecture of schizophrenia overlaps with other psychiatric disorders, and that some risk genes can predispose to different disorders depending on the other risk factors that are present. And STAG1 is also implicated in schizophrenia by common variants, which is interesting because we don't fully understand the roles of common and rare risk factors yet, and whether or not they act on the same biological pathways or different ones. And this would seem to suggest that some genes can be affected by both and that they can play converging roles. So this helps to inform our view of how rare and common variants contribute to schizophrenia risk. So to summarize, a new schizophrenia case control sample expands the number of cases available for analysis by 19%. This new sample is consistent with previous samples in terms of rare variant enrichment and supports the association of the 10 schema genes and the 32 wider schema genes. Meta-analyzing the sample with schema implicates 3 novel risk genes. And these novel genes support the overlap of schizophrenia genetics with other psychiatric disorders. And it also supports convergent roles of common and rare variation in schizophrenia risk. So thank you very much for listening. And I'd like to thank my supervisors, Elliot and James, the Psychosis Group, Mental Health Research UK and the Field Rose Charitable Trust, and the participants and their families. and I'm happy to take questions.